Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go. Let's make great science together.
Okay, let's unpack one of the most remarkable molecular mapping projects I've seen lately, the Stebula microcebus.
Right.
When scientists set out to map, basically every cell in the tiny gray mouse lemur, this primate that's like squirrel-sized, they found something really counterintuitive. It turns out the stem cells making sperm, the spermatogonia, their molecular instructions look more like immune progenitor cells than any other fast-dividing stem cell.
Wild.
Absolutely wild. And that deep link, this sort of conserved connection between stemness for making babies and stemness for fighting disease, that's really just the tip of the iceberg here.
Okay.
Our goal today is to really break down the key stuff from this huge nature paper, the Mouse Lemur Cell Atlas, and explain why this tiny animal might be a game changer for modeling human health.
So yeah, why the mouse lemur? I mean, it seems like a massive effort for such a small animal. We have mice, we have macaques. Why this little guy?
Well, it's about filling a really critical gap. Mice are great, easy to work with, breed fast, but, they're pretty different from us in key ways, like immune systems, metabolism.
Exactly. And neurobiology, too. Macaques are much closer, obviously, being primates, but studies take forever and are incredibly expensive. The gray mouse lemur, Microcebus murinus, hits a sweet spot.
It's tiny, right? Smallest primate, about 50 grams.
Yep. But the key thing is its generation time, just 8 months. Super fast for a primate.
Ah, okay. So you get the primate biology.
Right, which you need for studying things specific to us primates.
But on a timescale that actually allows for genetic studies, aging research, things that are just too slow or costly in bigger monkeys?
Precisely. And the scale of this atlas really reflects that ambition. Over 750 distinct cell types identified.
Wow.
From more than 226,000 individual cells across 27 different organs. It's a massive foundational data set.
Okay, 750 types across dozens of tissues.
Yeah.
That scale alone sounds like a technical nightmare. How do they even manage that consistently?
Yeah.
That sort of brings us to their basic method, right? What they show in Figure 1.
Yeah, they had to be incredibly rigorous. And they used a combination of sequencing strategies to get both breadth and depth. Think of it like trying to map everyone in a huge stadium.
Okay.
First, they used the droplet-based 10x chromium system.
Right, that's the one that does tons of cells quickly, the high-throughput method.
Exactly. That's like your wide-angle lens getting a basic profile of almost everyone or every cell in the stadium. That's where the bulk of the 226,000 cells came from. It gives you the sheer numbers, the breadth.
Got it.
But then they also use the plate-based SmartSeq2, or SS2.
And SS2 is more like a zoom lens, more detail on fewer cells.
Precisely. You zoom in on smaller groups, maybe just a few hundred or 1,000 cells, but you get much deeper information. You can read their name tags, basically.
Meaning you pick up genes that are expressed at really low levels, or maybe genes nobody even knew were there.
Exactly. That deep coverage is crucial for spotting those subtle differences that define specific subtypes, especially finding those unannotated genes.
So that dual approach let them nail down those, 768 distinct cell populations,
Yeah.
grouped into bigger categories like epithelial, immune, neural.
All the major compartments.
And did they find anything right away that screamed primate relevance? Like a quick payoff for choosing the lemur over the mouse?
They did, actually. A really clear example where the lemur immediately showed its value. They looked at stromal cells in the limb muscle,
Okay.
and they found adipocytes, actual fat cells mixed in. This fatty infiltration, it's pretty common in aging human muscle, often linked to weakness.
Right, I've heard of that. But you don't really see that in standard lab mice, do you?
Very rarely, if ever, in healthy mouse muscle. It's a significant physiological difference. So right off the bat, that finding suggests the lemur could be a much better model for certain aspects of human muscle aging or degeneration. It validates the choice.
Okay, so that's the who's who of cells. But cells aren't static. How do they capture the dynamics? Cells moving, changing over time or space? That seems like the next big challenge.
Molecular gradients, right? Exactly. This is where figure 2 gets really interesting. They found that many cells didn't just form neat little separate clusters. Instead, they formed these smooth, continuous gradients of gene expression.
Like a slope instead of separate steps.
Yeah, precisely. And these gradients often reflected either a physical journey through a tissue or a developmental progression over time.
The kidney example they use seems super clear for the spatial idea.
Oh, it's a beautiful example. The nephron is the kidney's filtering unit, right? The epithelial cells lining that tube formed this long, continuous molecular gradient, and it perfectly matched the known physical path. Starting in the proximal convoluted tubule, changing smoothly as you go through the loop of Henle, and ending up in the collecting duct.
So the genes a cell's expressing basically tell you its address along the tube.
Pretty much. It's like a molecular GPS coordinate within the nephron.
That's incredible. Does that level of detail give you new functional insights, like beyond just confirming the anatomy?
Absolutely, because you can see where specific genes turn on or off along that path. And they didn't just map the main filtering cells, they mapped the blood vessels woven around them, the vesirecta. And those also showed a smooth gradient, shifting from arterial markers to capillary markers to venous markers. Mapping these lets you pinpoint where specific hormonal signals are acting, how blood pressure might be regulated differently along the nephron, potentially revealing primate-specific kidney functions.
Okay, so that's space. What about time? Developmental processes.
They mapped those two, key lineage continuums. They could trace hematopoiesis blood cell development, showing, for instance, how stem cells branch off to become either neutrophils or monocytes and macrophages. And crucially, they mapped the entire molecular program of spermatogenesis in the male gonad.
From the very beginning, the stem cells.
Yep. From the spermatogonia, those initial stem cells, all the way through the various stages of development to the late spermatids, the nearly mature sperm. And mapping that pathway is what sets up that big, surprising connection we mentioned at the start.
Okay, hold that thought on the big connection for just a second, before we get there. Figure 3 seems to highlight just how much new stuff they found, right? Hidden diversity.
Oh, definitely. The atlas didn't just confirm what we thought we knew, it uncovered dozens of previously unknown or only vaguely described cell subtypes.
Really? Like what?
Well, there was way more diversity than expected. In cell types we thought were fairly uniform, like fibroblasts, the connective tissue cells, and macrophages, the immune cells. They showed huge differences depending on which organ they were in.
Interesting.
But let's zoom in on the liver, the hepatocytes, the main functional cells.
Okay, they found two types there. CPN2 high and CPN2 low. Why is that a big deal? Don't we already know liver cells are arranged in zones?
We do, but these two subtypes didn't neatly map onto those known zones. What was interesting is that one type, the high type, expressed way more total RNA. And the thinking is this might correspond to a really fundamental difference in liver cells. The larger ones that are polyploid, meaning they have multiple sets of chromosomes, versus the smaller diploid ones. Ah, okay.
And the fact that this molecular distinction seems to exist in humans, lemurs, and mice suggests it's something pretty ancient and important for how the liver works.
That makes sense. And wasn't there also something about specialized capillaries, like really specific jobs for tiny blood vessels?
Yes, the FABP5 plus RPP7 plus capillary subtype. A fantastic example of specialization hidden in plain sight.
What did they do?
These capillaries were packed with genes involved in grabbing and binding fatty acids. They seem specifically geared towards energy storage or handling fats.
Where did they find them?
Mostly in tissues with really high energy demands: the heart, the kidney, active muscle. It totally reframes how you think about capillaries. They're not just passive pipes. Some are highly specialized workers.
Okay, fascinating detail. Now let's zoom back out. Figure 4, the global comparison. This is where that initial hook comes back. The immunity and reproduction link.
Exactly. So they took all 750 or so molecular cell types they'd defined and did a massive comparison, looking at overall similarity. How do you even compare that many types globally? Do you average the cells?
You got it. They create what are called pseudo-bulk expression profiles. Basically, you average the gene activity across all the cells you've defined as belonging to one specific type.
Okay, so you get an average profile for each cell type.
Right, and then you compare those average profiles. And when they did that, this striking pattern emerged. The male germ stem cells, the spermatogonia, they clustered most closely with the immune progenitor cells. Not other rapidly dividing cells, not other stem cells, but specifically immune progenitors. They shared a unique molecular neighborhood.
Wow. So reproduction and defense. Sharing a fundamental molecular signature at the stem cell level, what genes did they actually share?
Well, they shared enrichment for certain genes involved in the M phase of the cell cycle, things like CCNB1, VRK1. That suggests some similarity in how they handle the mechanics of division.
Okay, that makes some sense for dividing cells.
But crucially, they also shared enrichment for genes not directly involved in the cell cycle, like Tessmin and RSPH14. And that implies these genes might be part of a deeper conserved program regulating stemness itself, that ability for indefinite self-renewal that both sperm production and the immune system rely on.
That's a profound connection.
Yeah.
Okay, so this atlas isn't just a catalog. It's revealing fundamental biology. And it really seems to solidify the lemur's place as a key model, right? How did they quantify that advantage over the mouse? That's section 4, the evolutionary part.
Exactly. They did a direct comparative transcriptomics analysis. They picked 63 cell types that clearly had counterparts orthologs in human lemurs and mice.
Okay.
And they looked at about 13,000 core genes that also have direct one-to-one orthogs across these species. Then they basically asked for a given cell type, does the human gene expression pattern look more like the lemur's pattern or the mouse's pattern?
And the answer was?
Overwhelmingly the lemur. The correlation, the similarity between human and lemur cell types was almost always significantly higher than between human and mouse.
So statistically, the lemur is just a better molecular match for the equivalent human cell across most of the body.
That's what the data shows. It provides quantitative validation for using the lemur as a proxy for human cell biology in many contexts.
But it wasn't the same across all cell types, was it? Some parts evolved faster than others.
No, definitely not uniform. Evolution tinkers differently in different areas. And guess which cell types showed the biggest difference, the biggest advantage for the lemur model compared to the mouse?
Let me guess, based on earlier discussion, the sperm cells.
You got it. The developing male germ cells, especially in the later stages of spermatogenesis, that pathway showed the largest molecular divergence between mice and primates.
Meaning that process has evolved really rapidly since mice and primates split off.
It seems so, which makes a primate model like the lemur even more critical if you want to study the specifics of human sperm development or fertility.
Okay, so the practical upshot of all this comparison is this list they created, the HL genes.
Yes, the human lemur conserved genes that are divergent in mouse. This list is pure gold for researchers.
Why is that?
Because it flags genes and pathways where the mouse model is likely to be misleading for human biology. Think of it as a warning label. Caution. Primate-specific function likely here. They found genes like SFRP5, PLCB3 that fit this pattern.
And the clinical relevance.
Get this. About 26% more than 1 in 4 of these HL genes are already linked to known human diseases through genome-wide association studies or other genetic links. So this atlas immediately points researchers towards specific disease pathways where using a mouse might give you the wrong answer and where the lemur is probably the model you need.
That's huge. It lets you focus resources where they'll have the most human relevance. Okay, so stepping back, the innovation here isn't just one discovery, it's the whole approach, isn't it?
Yeah.
Applying this massive, standardized single-cell technology to, well, an unconventional primate model.
Absolutely. That standardization is key. Pulling off an atlas of this quality and scale across so many tissues with consistent methods through a big international group, the Tabula Microcebus Consortium, that sets a benchmark.
Right.
It demonstrates how to systematically build these foundational atlases for any new model organism we want to develop.
And for the lemur specifically, this atlas is now like a scaffold, right, for future work.
Totally. If you're studying things where primates differ significantly from rodents, maybe complex neurodegenerative diseases, maybe sleep patterns, metabolism, certain aspects of aging.
Areas where the mouse has sometimes been tricky.
Exactly. You now have this incredibly detailed molecular map for the lemur. You know which cell types are involved, and critically, you have that HL gene list telling you which genes are conserved between humans and lemurs, but different in mice.
So you can design genetic experiments, maybe using CRISPR or other tools in the lemur, targeting those specific HL genes linked to a human disease, feeling much more confident that the results will actually translate.
Precisely. It dramatically increases the efficiency and potential relevance of translational research using this primate model. Less guesswork, more targeted investigation.
Okay, so to wrap up, a massive cell atlas for the mouse lemur, revealing incredible cellular diversity, finding unexpected molecular gradients, uncovering that surprising link between immune and sperm stem cells, and really quantifying why the lemur is a better model than the mouse for many human-relevant genes and pathways.
A huge achievement. And, thinking bigger picture, the paper mentions that the lemur group, the Lemuroidea, is incredibly diverse. Over 100 species have evolved.
Yeah.
And we just discussed how the late stages of sperm production seem to be evolving particularly rapidly, molecularly speaking, at least across the species they looked at.
So you're connecting rapid evolution and reproduction with rapid speciation.
It raises a really fascinating question, doesn't it, for you listening, to think about, could this fast evolution in reproductive cell types, maybe driven by things like spring competition between males, actually be one of the key engines driving the diversification, the speciation within primates? It's the kind of big evolutionary question that these incredibly detailed molecular maps can suddenly bring into focus.